Hello class, welcome to your first online lecture, and in this lecture we're going to talk in more detail about the nature-nurture controversy, which is something that I brought up in the first class and we've talked a little bit about in the second class. And so in this lecture we're going to really try to address this misconception head on. And that's why I put verses here in quotes. Nature versus nurture. Is that really what we want to be thinking about? And for some reason, people in the world still want to divide the features of living organisms or traits into those that are caused by nature or genes and those that are caused by the environment or nurture. This controversy, as we talked about the first day of class, is really old, but unfortunately it's still an ongoing debate and discussion. And I really want you guys to understand the scientific basis of why this viewpoint is so seriously flawed. Okay, so in popular view, a lot of times people think of instincts as genetic and learning as environmental. And in reality, this idea is just wrong. No. No. It's just not true. Really, what we want to consider is the idea of nature and nurture, right? Not or, not versus, but both. And what I really want you to think about is what we consider gene-environment interactions. We want to look at the interactions between genes and environment. Okay, let's go back to a few words that we just were learning about. And one important word is the idea of phenotype. What is a phenotype? And remember that the phenotype is something that you can observe, those observable characteristics of an organism, and that those characteristics or traits can be physical kinds of characteristics, they can be behavioral, they can be chemical. Mostly we're thinking about behavior and physical features of organisms that we can observe. And the phenotype then is controlled by two main things. What phenotype you have, let's say if you're a ladybug and you have red wings, or if you're a dog and you have the ability to retrieve a ball, that's a behavioral phenotype, retriever phenotype, that phenotype is controlled both by genotype and what? I'm pausing here a second so that you guys can all sort of yell it out loud. Phenotypes are controlled by a combination of genotype, which are the genes that you have, plus the environment. Right. Glad that you all got that. Genotype plus environment. And so every phenotype, every single one, like we talked about before, every single phenotypic trait is controlled by a combination of two. And again, the relative influence of genes versus environment can be different, but both are always involved. Okay. So let's move down to thinking about what we mean by phenotypic variation. Remember that we talked about populations being composed of lots of different individuals, and that those individuals vary in what they look like or how they behave in their characteristics. They differ across the individuals within a population. They differ with respect to their physical and behavioral traits. For example, you can have a whole bunch of dogs that behave very differently and look very differently. You guys are familiar with lots of different dog breeds. Well, those animals can interbreed. They're the same species. There's tremendous amounts of phenotypic variation among dogs in the world for a lot of different reasons. There's lots of phenotypic variation, say, among lady beetles. You can count the number of spots and see that they're different. There's phenotypic differences among humans, as we've already talked about, in terms of not just how we look, but how we behave. And so that phenotypic variation among the individuals of a population is due to both genes and environment. Okay? And so when we're thinking about how much variation there is in a population, there's a tremendous amount of variation because of the fact that either genetic differences among individuals are going on, or environmental differences among individuals are going on, or a combination of the two. So sometimes you see that genetic differences among individuals can have a greater influence, and sometimes the environmental differences that individuals are experiencing have a greater influence. Okay, so for example, those differences in the physical features of dogs that you might be observing, that there can be definitely genes that are determining whether your dog looks like a poodle or whether it looks like a German Shepherd. Okay, but other attributes of those dogs in terms of how they're behaving and what they do and other features that they look like, they all have teeth, they all have a tongue, they all have ears. There's a lot of similar genes among those organisms. Okay, and that the way that they differ can be a lot attributed to the different environments that they've experienced, where they were raised. Okay. Okay, as we consider how genes and environment are influencing phenotypes of individuals within populations, we have to remember that when genes and environment are interacting, this interactive process that I mentioned before, that this is happening throughout the lifetime of an organism. So it begins with when organisms are first conceived, if you're talking about sexually reproducing organisms, and continuing all the way through adulthood. So the development of an organism is in itself an interactive process between genes and environment. Let's look at some components of this, and there's kind of a bit of text here, but I think you'll get it as we go. All right. So as an organism develops, the genetic information that's in the cells of these organisms is interacting with the environment that's changing. Okay, so there's both an internal environment inside the body of the organism, inside the cells of the organism, between the cells of the organism. So there's that internal environment as well as the external environment, what's outside in the world. Okay, that genes can be turned on or off by certain signals. This is critical. I think a lot of people don't realize this, that there are genes that you might have that will never be expressed. I've mentioned before that many people have cancer genes, but because those genes are never turned on, that they never get cancer. And in fact, it's believed that most of us, or almost all of us, have certain genes that could potentially cause us to have cancer. But fortunately, those genes are never ever turned on. Okay, so the signals that are turning genes on or off can be these internal cellular chemical signals, things like hormones that I've mentioned before, as well as enzymes, other kinds of internal chemicals that can be in the nucleus of cells, that can go from the cytoplasm into the nucleus of cells, or they can come from the external environment, and signals like light, like sound, like temperature, and even more complicated kinds of environmental stimuli can influence how genes are functioning. Finally, these interactions that are going on as an embryo is developing into a fetus, eventually into an adult, okay, during the whole developmental sequence, there are interactions that are happening during the assembly of the organism as it's developing, as the neural networks are coming into place, as your brain is forming, as your spinal cord is forming, as the nervous system is forming, as well as other aspects that have to do with your anatomy, not just your nervous system. So the interactions, the gene-environment interactions are going on and determining how your neural networks are getting set up to start with. Does that mean they're going to be completely fixed in place? No, because those interactions are going to continue later too. But these early interactive processes can determine a lot of how an organism is going to be manifested. Let's look at the example of honeybees as we're thinking about how organisms change throughout their developmental history. So if we think about worker bees, honeybees, many of these kinds of things, hopefully you're already familiar with, that early on bees start out as, they start out as larvae, but when they hatch into workers, they become nurse bees. And then over time, they transition later in life to foragers. So the nurse bees are those bees that are inside the hive. They're doing the grunt work. They're cleaning the cells. they're feeding the newly developing larvae from new eggs that are laid and hatching into larvae. They feed the nestmates, the ones that have already hatched and have come out. Eventually, these bees start to leave the nest as they get older. You can see the transition of age down here, right? So we're going from 0 to 25 days. So eventually they leave the nest and start gathering and packing up pollen and foraging outside in the real world. So not to be confusing, that the packing of pollen happens from the pollen that's already brought back, and then the foraging is going out and getting nectar and getting pollen to be packed once they return to the hive. So we can ask the question, does genetic activity in a honeybee shift as it's going from nurse bee to forager? Is this change of behavior, like you're not cleaning and taking care of stuff anymore, you're going out in the world and you're foraging? Are there genes that are responsible for that, or how much are genes responsible for that? So this is kind of a complicated diagram, and you certainly don't need to be responsible or think about all of the details of it. But I basically want you to look at these two columns on the left here. I'm going to just kind of circle them. We're going to ignore the 4 little columns on the right. Those are the results of some interesting experiments that I don't have time to describe to you. But so we have nurses that are along the left side here, right? And we have foragers on the right. Kind of making too much of a mess with this pen. But anyway, when we ask the question, do changes in genetic activity go? This results of this what's called microarray technology. It's a molecular biology technique that you can look at relative levels of gene activity in a really large set of genes. And in fact, in this particular study, they were looking at the activity of about 5,000 or more than 5,000 genes. And so here we're just looking at about 17 different genes of those 5,000. All right, but if we looked at all that data, you would really see the difference even more dramatically. But what you can tell is that if it's yellow, there's a really high gene activity greater than 2. If it's blue, there's low gene activity. So what you can see is that for this set of genes over here, nurses have high output of these kinds of genes, but foragers don't. Okay, and for this set down here, nurses have very little gene activity that's going on, meaning that these genes are off. Okay, and that in the foragers, it's these genes that are on. Okay, so the second set goes on in the foragers, and it's the first set that's on in the nurse bees. Okay, hopefully that makes sense. And basically what I'm trying to illustrate here is that yes, there are changes in many, many genes that are going on in this shift from nurse bee to forager bee.